biogenesis is basically what we are going to talk. And one of the key items that I want us to be able to look at carefully now is the definition of key terms. And in the definition, maybe we'll be looking at a few things that are quite very important when you're talking about microbial pathogenesis. One of the next area that we look at is the barrier system of the body. How does the body able to protect itself against some of those infections? Like we're talking about antibodies, you're talking about the cell of the immune system and such a things. When we're talking about Koch postulate, basically what we are talking about is what principles are there that proves that the disease are caused by infectious diseases. Then you look at stages of pathogenesis, types of pathogens, and specifically we'll be going to specific pathogens so that you gain the knowledge and be able to know what to do. Definition of key terms, for example, we look at pathogenicity. Pathogenicity, the ability to cause disease, of course, and then pathogenesis, it is a process. Refer both the mechanism of infection and the mechanism by which disease develops. And then virulence factors, degree of pathogenicity. For example, we look at a very simple example of some of the bacteria that are there. For example, you can compare the virulence. Can you compare the virulence of cholera and the virulence of Escherichia coli? As much as all of them cause diarrhea, but one causes more severe form of diarrhea than the other. And then the other term is infection. Infection is growth and multiplication of microbes in or on the body with or without the production of disease. And then the capacity of bacterium to cause disease reflects its relative pathogenesis, like I talked about, Escherichia coli and Vibrio cholerae. Both of them cause diarrhea, but one causes a severe form of diarrhea than the other. Then another task I'll give you, meanwhile, is for you to look at colonization, contamination, primary pathogens, and opportunistic pathogens. Let's look at the barriers system of the body. And the first barrier, of course, is the skin. And the skin, you have some mucus, and some of us need to have the melanin. And you realize that don't have melanin, and those that have melanin, their barrier systems in terms of strength is never the same. And then you're talking about innate immunity, what you're born with, the type of immunity that everyone is born with. Like we talk about inflammation, the complement, the macrophages, cytokines. These are specific chemicals and specific processes that are there to ensure that the body is really protected. So like then we talk about adaptive immunity, is the immunity that comes as you get exposed or maybe you are vaccinated. So we have antigen specific B and T cells, and then we also have other type of immunological response such as antibodies, that is humoral immunity, that helps us to be protected against some of this infection. Host defenses can be compromised by destructing barriers or defective that results to defective immune response. For example, somebody who is HIV positive, somebody who has AIDS, the immune system is never the same as the other one. So let's look at briefly the Koch postulate and what it really means. It just tries to tell us that if you are sick, for us to say that you have a communicable disease, it is quite a simple process. Let's look at a healthy organism, and then you get the red blood cells. Extract all the red blood cells, and then go and culture the red blood cells. Do we get anything out of it? If we don't get anything, then indeed there's nothing that might have been a problem with the rat. But let's look at a rat that has died, and then you get the red blood cells, and then we go and look at the microscope. You able to see later on that probably there's some microorganisms there. For example, if we look at somebody who has malaria and then is sick, of course, if you get the red blood cells from that person, you'll be able to say that that person has malaria. But what about if somebody has bacteria? Bacteria in blood. Bacteria in blood is simply called bacteremia. So we're able to culture the bacteria. So if we go and grow it, grow that bacteria in the culture, and then take that same bacteria that we've grown in the lab, and then inject it to the healthy bacteria, a healthy rat, what will happen? Definitely, we expect something to happen. The rat will get infected, and then it will die. So let's look at stages of pathogenesis. The first stage is exposure. Before we say somebody is going to be sick or is going to get infected, then you have to say that you must have gotten an infection. You encounter a potential pathogen. For example, now, the first exposure now, maybe you get coronavirus. Then that would be a very good example, because currently we are, and I'm having this lecture simply because we cannot have our class in normal class simply because there's coronavirus. So we will not want to expose you. I will not want to be in contact with you. Probably I might have coronavirus. So then for coronavirus to be able to be in you and grow in you, it has to adhere. And that is called adhesion or colonization. For example, it has to grow in your lung. It has to adhere to the lungs and escape the brushing of the lungs and the mucous membrane. So you don't cough it out. Once you've coughed it in, it is inside now. So it's going to stick in you. Then we're talking about invasion. Invasion involves dissemination of the pathogen throughout the local tissue of the body. So you've just inhaled this in your nostril now. It has now going down the lungs. And that is where it's going to cause problems. Then the next stage is infection. A successful multiplication of the pathogens leads to infection. Infection can be described as local, focal, or systemic, depending on the extent of the infection. Pathogen must first gain access to the host, and that is a principle, and must either penetrate before infection is established, then must continuously evade host defenses. We are talking about host defenses, that is in terms of the antibodies that can be able to kill the infection, the mucous membrane that it should be able to actually help it to stick there, and then maybe you sneeze it out. And infection usually causes host damage at the end, like cause lung problems, cause fever, and cause anemia, and cause stomach ulcers, as in the case of H. pylori. So to cause disease, a pathogen must gain access to the host, adhere to the host, penetrate and evade the host defenses, damage the host either directly, and accumulation of the microbial waste into the host. And that is quite a very big problem. So let me remove my face so that it looks there. So now when you're talking about that, basically you're talking about how does disease come into being, and those stages must always be followed. Sorry for the phone. Pathogenic properties of viruses. Let's look at viruses in a very simple way. Of course, we'll talk about bacteria later on, but when you're looking at viruses currently, and that means that the mechanism to evade the host defense mechanism is the first thing, because it should be able to evade that so that it grow inside. For example, one of the mechanisms that is used to evade the host defense mechanism is growing inside the host cell to hide from the immune defense. And then that is one very big challenge, because once it's inside the cell, then the immune cells that are going to be looking for this infectious agent may not be able to do so, may not be able to find it, and then kill the immune cell. For example, the HIV cell that kills the T helper cells. So if the immune cells are killed, and actually they're the ones that are actually very important in trying to help you to protect you against that infection, then the HIV will continue growing because there's no way that the immune cells will be able to be there because they're already killed anyway. So one other way we will look at is cytopathic effect that comes with the viruses. Cytopathic effect is the visible effect of the virus infection that are out there. That some of the effect that will kill the cells, some will change the cell. One of the cytopathic effect is stopping the DNA, RNA, or protein synthesis, such as herpes virus that block mitosis. So that one is a cytopathic effect because cyto means cell, and then pathic means the effect, the unwanted effect, the disease effect, the deadly effect that will happen. So let's look at another cytopathic effect is lysosomal autolysis of the host cell, e.g. influenza, that causes the bronchiolar epithelium to really be able to um, die in the end of the day. Production of the inclusion bodies. Inclusion bodies are visible viral part inside the cell that can identify a particular virus, such as rabies virus or negri bodies. This syncytium formation, neighboring cells fuse together to form bigger cells. And this is a very common problem, especially in RSV and respiratory syncytial viruses, that cause all the neighboring cells to fuse together to form a mass that will really cause a big problem, especially in the respiratory system. Change in cell function. The cell will not function normally. Production of interferons by the host. So interferons actually trigger the host immune response, and some of the host immune response at a certain an infected state is really a very has a detrimental effect to the cell. Induce antigenic changes of the host cell surface. So this one is quite a very important factor that if it causes the trigger of destruction of the infected cell by the host immune response, and that one means that it does not directly kill but induce other cells of the immune system to kill that very cell that are very important in protecting against such infection. Induce chromosomal changes. So chromosomal changes means that the cells chromosomes have changed, and probably will be one of the effects that we will not like in the end of the day, because if the chromosomes change, then they may be able to change in a very bad way, like causing cancer cells. So the cells have an ability that make them to grow in a very abnormal way, and in the end may cause cancer. And the same thing like cell transformation. At that cell transformation, this may activate or deliver oncogenes, resulting in loss of contact of inhibition. So that is cancer. So the cells continue to multiply abnormally. And one of the big example is the papilloma virus. So let's look at the fungi. How do fungi cause the disease? They produce toxins causing allergies, and for those nutritionists to be able to appreciate this, and maybe when you be reading more, or you'll be able to realize that fungal toxins are quite a big menace, especially in the era of food production and food preservation. Stachybotrys causes headache, vomiting, and mental disturbance. Quite a very big problem. Invasive systemic mycosis in immunocompromised patients, such as Candida. Mushrooms also have mycotoxins that may be hallucinogenic or deadly. For those of us who go and forage for mushrooms, you might eat some of the mushrooms that are not really very nice and are really poisonous for you. Let's look at protozoa. Briefly, I'll talk about protozoa. I've jumped so many parasites, but we'll be able to talk about them in the later discussion. Protozoa can grow inside a host cell, causing lysis. For example, malaria, the plasmodium. So that means that the cell, the red blood cells that the parasite is growing inside it, they die. They get destroyed, and some of these cause very big problems such as anemia. So they use the whole cell as food source and produce waste that cause diseases. So let's look at algae. It's not a popular thing, but some of us who are living in the coast will be able to come across shellfish poisoning, which is quite a very big problem, especially for those of us who consume seafood. And that is the end of my discussion for today. For those of us who will have questions and maybe you want clarification, maybe in the later presentations that I'll be able to put, then we'll be able to discuss more. And I'll encourage you to ask me questions on MLM so that we can be able to help each other understand. This is an introduction and it's also my first long presentation, and I believe you'll have learned a lot today. Bye. Have a nice day.